Well, first of all, thank you for agreeing to this video interview for BioNews.
My pleasure, thank you.
Thank you. Okay, so we have here Arndt Rolfs, CEO of Centogene, and we wanted to talk to you a little bit about your presentation this morning and about your company's philosophy and its approach to rare disease.
Thank you.
And for those people who are just joining us and watching the video, could you just maybe summarize your presentation and your short talk in maybe a couple of paragraphs?
Yeah, very happy to do that. So Larry, thanks for that. Centogene is dedicated to improve the early diagnosis of patients suffering from rare hereditary diseases. We're all aware this is a huge effort in the future because we can speculate that there should be at least 350 million people suffering from any form of a rare hereditary disease. However, still the diagnostic delay in the majority of the patients is about 10 to 15 years. And the later we are diagnosing the patients, the more we can expect that there is a progress of the disease with irreversible damage of the organs. Therefore, a must to improve the overall prognosis of the patient is definitely improve the early identification and diagnosis. This is where Centogene is dedicated to, and it's very clear this has to be a global activity, because patients are not only sitting in the US or not only sitting in Europe, especially the autosomal recessive inheritance mode is due to the consanguinity, especially in some Arabic countries in North Africa, in India, Indonesia, Malaysia, causing that some of the diseases are up to 10 to 40-fold more frequent to whatever we know from the European environment or the US American environment. Centogene is therefore active in more than 100 countries. And the majority in Middle East, Latin America, Canada is an extremely important country for us, Europe, India, Indonesia, and Malaysia. And in these countries, we are offering either a clear-cut genetic-based diagnostic approach. That means we are getting a sample from the patient, we are analyzing, sequencing the sample, and we're coming back with a result. However, in that approach, it's always the difficulty to properly interpret the data, because for ethnicities others than the Caucasians or the Europeans, we are still at the beginning to understand what DNA makeup can cause the different diseases and whether a specific mutation in such disease is really causing the phenotype in our patients. That's one of the reasons why Centogene is also very active in the second layer of the technology, and this is the proteomic and metabolomic approach. Because it's the proteomic and metabolomic approach.
Could you explain that?
That's very simple. The genomic approach is just analyzing the gene or the different genes within the genome. We call the entity and complexity of all of the genes the genome, and therefore the technology is called genomic. And the same is true for the proteins. So it means for all of the proteins we know in the human body, we call them the proteomics, especially if we analyze these proteins. And if we're talking about the metabolites of the proteins, then we're talking about the metabolomic. So you can also label this as a multi-omic approach, which means combination gene, protein, and metabolite. The nice story around the proteomic and metabolomic is the fact that this is the readout of the DNA. Because the DNA will be transcribed in RNA, the RNA will be translated in protein, and proteins will be typically degraded or metabolized in metabolites. Whenever we have a functional mutation within a specific gene that causes the damage of the protein, you can demonstrate this on the protein level. And therefore the proteomic approach, which means the analysis of all of the existing proteins, demonstrates you always as a readout the functional consequence of a specific mutation.
Oh, just a question. Your corporate approach. You're about a 45 million euro company. You mentioned that you're present in 100 countries. You also said that a lot of these diseases have been studied only in the United States and Europe, basically among white people, among Caucasians. The vast majority of the world's population are not Caucasian. Yep. So how do you reach that 90% of people who have never really been in clinical trials, never been covered?
Yeah, that's a good question. I guess extremely critical, and that brings it exactly to the point where we have to improve the entire diagnostic process. Typically, diagnostics means you have to have access to specific technologies. And then it's a question who's paying for the technology. It's the topic of reimbursement.
Right.
Therefore, there's clearly a bias that the majority of the patients actually be known and been diagnosed coming from the European, from the US American, maybe from the Japanese environment, maybe from Israel, maybe a little bit from the Middle East, and that's it. Therefore, the most important element in how to speed up the early identification
say that again, please.
The most important element how to speed up the early diagnosis and identification of the patient is simplicity of the diagnostic workflow. If this is a complex workflow, if this is an extremely expensive workflow, it's not working in countries like India, Indonesia, Malaysia. Therefore, you need a simplified, stable, robust workflow. And that's exactly the reason why we at Centogene believe that it will be not the genetics in the future that will allow us to simplify and speed up the early identification. It's most likely around the proteins and the metabolites that can be so nicely measured in the meanwhile based on a technology called tandem mass spectrometry. Tandem mass spectrometry, once it has been established, is extremely robust, highly sensitive. We are in the meanwhile in the upper femtomolar concentrations. We can detect in the peripheral blood. And the nice element in the tandem mass spectrometry is as well that you can multiplex it, which means you are not only measuring one target, not only measuring 5 targets, you can measure 500 targets or 5,000 targets if you have characterized the individual target a little bit like a fingerprint for a specific disease. Then it means you are generating a spectrum of 5,000 different peaks in the mass spectrometry, and you know exactly peak number 1 represents disease A, peak number 2 represents disease B, and so on. And therefore, within a simple screen, you get with a high sensitivity information for several hundreds, ideally several thousands of diseases. And that simplification and dramatic price reduction will give us really the chance to have a breakthrough to get these patients also diagnosed in not so very wealthy countries.
Very interesting. Is anybody doing something similar to Centogene in this regard?
To the best of my knowledge, we are the only company worldwide combining this genomic with proteomic metabolomic approach in the routine diagnostic setting. Research, there are a lot of activities, but in the routine setting, to the best of our knowledge, we are the only one, number one. And number two, I'm pretty sure we are definitely the only one doing this systematically in thousands, 10,000s of patients to build like a library regarding the information of the different peaks and the different interpretations of the mass spectra from the tandem analysis.
You mentioned a dramatic price reduction. Can you give an example of what your technology costs per patient versus what's being done now?
I'm pretty sure in 5 to 10 years we are talking about analysis in the range of, let's say, $5 to $10. Definitely not more. Because once the technology has been established and validated, then you can easily run it, and there are no expensive chemicals necessary, no expensive consumables necessary. You have only to pay once for the machine, for the system, and then for the maintenance costs. But the analysis itself, extremely cheap.
For example, let's take the issue of newborn screening. As we discussed last time when we met, only a few states have mandated mandatory screening for spinal muscular atrophy. Yeah. I think 5 states and then another 12 states are considering it.
Yeah.
Why isn't everybody testing for diseases like SMA?
Yeah, I guess there are 2 answers per today. We are not yet at the stage that we can measure all of these diseases with extremely cheap technology like the mass spectrometry. But let's think in the history. The reason that the newborn screening developed so nicely in the 70s and 80s was just based on the fact that mass spectrometry was introduced to the newborn screening. And this was for sure in the primary point focusing on the metabolic diseases, because the metabolite can be very easily measured in the peripheral gut. SMA is not a metabolic disease, therefore you need a little bit of more complex technology to really prove whether, for example, there is a specific deletion of a specific exon within the gene. You can't do this per today in the routine. Research is always doable, but you can't do this in the routine per today with the tandem mass. So it's a little bit a price topic, but I guess there is more, and that means whenever we are talking newborn screening, there should be ideally always a therapeutic consequence of the screening.
There should what?
There should be always a therapeutic consequence for the
Meaning
the
that if a baby is found to have the disease, you better
should be
have a
treated.
way to treat it.
Oh, absolutely. It started with PKU, as you're aware, in the 50s. Then we added the hypothyroidism, and the reason for these two diseases was just based on the fact that there was just a cheap and robust treatment available.
Right.
For PKU, the diet. For the hypothyroidism, it was just a substitution of the missing molecule for the thyroid. In nowadays, treatment is getting more expensive, especially for the very rare diseases.
Right.
And I guess that's one of the reasons why governments are a little bit hesitant to allow this very nationwide screening for the more rare disease where the treatment is more expensive. So it's a really a social topic. Are we willing to spend a significant amount of money in the early treatment of the patients? And there are some predictions, as you're perfectly aware. If we are moving forward in the same way as we have done in the last 10 years with the development of orphan drugs, in 10 years we are spending 14 to 15% of the social money for the healthcare system just for 0.001% of the population. There is some disbalance, and this is a topic that has to be solved. And this is extremely critical that we are starting to have a very open discussion about that. Otherwise, I would personally fear that there will be still the politically driven or the governmental driven intention to stop the early identification of the patients and to avoid that the patients are early enough diagnosed.
Well, just the question though, if you don't diagnose and a baby grows and is found to have SMA, then the cost of treating that kid is also going to be astronomical, right? If you treat early, you might actually, with Spinraza, for example, you might actually cure the child.
Absolutely with you. But this means you have to change the mindset. That's not always following rational arguments, these decisions. And let's not forget there are for sure also some religious intentions sometimes in discussions like these, where based on the religion, some people might argue this is not something where we should interfere. So there are a lot of mixture and weird ideas, not always following rational and scientific arguments. I'm totally with you.
Right. You mentioned that you're doing a lot of work in the Middle East, particularly in the Gulf. Mm-hmm. Can you talk about why you're focusing on Arab countries?
Oh, that's rather easy. As I already mentioned, autosomal recessive diseases are reflecting roughly about 70 to 75% of all of the rare hereditary diseases. So the overall majority of these diseases are following the autosomal recessive inheritance mode. In these areas, Arabic countries, Turkey, Saudi Arabia, Kuwait, UAE, specific parts of Pakistan, India, Indonesia, Malaysia, we have a high level of consanguinity. It means marriage within the same family. That always results in a higher likelihood that you will say in the next generation an affected member of the family, typically a newborn or a child. Therefore, the frequency of the autosomal recessive disorders are 10 to 40-fold higher compared to the Caucasian or European, US American population if you're going in these geographical areas.
10 to 40.
10 to 40-fold higher. And since we are interested as a company to generate knowledge in the sense of better understanding the phenotype, better understanding the genotype-phenotype correlation, for sure those countries are more attractive for us because we are identifying more patients on the one side. And let's be fair, it's also a commercial element that we are getting easier paid for the diagnosis of these patients, whatever we are doing, in contrast to the somewhat a little bit weird reimbursement system in the US.
Could you briefly touch on this issue of Spinraza? And the reason is that we understand that within a matter of days or weeks, the FDA will approve the new Novartis gene therapy, Zolgensma.
Yeah.
If a gene therapy becomes available for something, a disease like SMA, which obviously you're involved in, would that somehow not only bring down the price of an existing therapy like Spinraza, but would it somehow make it irrelevant over the long term? if it's cheaper to have a one-time gene therapy rather than spending $300,000 to $75,000 every year, wouldn't that be the way to go?
Yeah, I'm totally with you, but let's be fair. On the one side, actually, no one has really long-term prospective data regarding the efficacy of gene therapy. We all believe in the huge future of the gene therapeutic trials and the borders, but no one of us has ever seen patients after 10 or 15 or 20 years being able to demonstrate that the gene therapy is still working. So there is some belief or speculation in the future, number one. Number two, if you're not sure how long you can expect to see really a curement for the individual patient, would you really be willing to pay at the beginning of the treatment $1.5 million, $1 million for the treatment? Again, it's a speculation in the future. Therefore, I'm totally with you. Once we have better scientific data for the long-term treatment, and we can really prove that the patient, once they are getting once in their life a treatment, they are stable for several decades, then the gene therapy will definitely the treatment of choice compared to a lifelong treatment with a higher expensive price. Totally with you.
I think that wraps it up. I really appreciate your time.
Thank you. It's my pleasure.
Good.
Thank you.